Welcome to today's webinar, Whole Slide Quantitative Immunofluorescence on Brain Session Made Simple. Today, we are going to talk with some keynote speakers, Charline Baraban, and from the customer review, Erik Castillo. My name is Amarlig Ferrigni, and I am product specialist at Paralab Bio, Spain. I am talking on behalf of my colleague Andrea Zapater from Spain also, and from my colleague and Alexandre Borges from Paralab Bio, Portugal. We know your time is precious, so thank you for joining. I'm very happy that you all are here. Today we'll discuss how the whole slide quantitative immunofluorescence can be simplified, accelerated, and made more reproducible, particularly in complex tissues such as brain section. We are pleased to host two excellent speakers who will guide us through both theoretical and practical aspects of this workflow.
Hello.
Hello.
Okay. Before we begin, just a few practical details. The session will last around 60 minutes with the including live Q&A session at the end. Please feel free to send your question at any time using the chat. During the session, in the right side, we will share brochures and application notes that you can download. This session is being recorded and will share the main resource afterward. Please feel free to send your question at any time. Here is the agenda for today. I will begin with a short introduction to Paralab Bio and then the learning objectives of this session. Then Charline Baraban from Innopsys will talk about technical overview of InnoQuant, and from workflow experience, Erik Castillo from Translucence Biosystems, a USA company. At the end, we will have a Q&A discussion. Let me start with a brief introduction of who we are in Paralab. Paralab is a company that was founded in 1992 by two PhD students in chemical engineering with a strong scientific and technical background. They have like this spark to do something different, and they create Paralab. Nowadays, Paralab have so many divisions, so some divisions like Paralab Bio, Paralab Engineer, Paralab Green. Our headquarters is in Oporto, but we also have office in Lisbon, Barcelona, Santander, and very soon in Madrid. We have some certification that like ISO 9001 since 2011 in Portugal, and recently in Spain, 2006, we have this certification in management. Also, we have a laboratory that I will show a picture in the next slide that has GMP certification since 2022, quality control contract analysis, R&D license for medical cannabis, that is for Paralab Green, since 2024. Also, we have an ID seal since 2025 recognition of suitable for conducting research and developing activities. This is our main achieve, one of our main achieve in the office of Oporto, that we have in some applications in the laboratory, and we can provide analytical service to research in analytical chemistry, to pharmaceutical instruments and industry and material science. These are our 3 points vision: mediating the process of supplying and maintaining scientific instruments and equipment, offering technical consultancy and training, and providing analysis services for material characterization. Also, we are very proud to contribute to scientific and knowledge dissemination. One of the, also our very proud that we are a strong team formed by 20% with people that have a PhD, 10% with people that have master in science. So we have a strong team that can help our customer in with a lot of a great background, scientific background. In the dissemination of the information, we go to a lot of events like Congress. Here are some pictures from the last year with my colleague Andrea Zapater and some from Alexandre in Portugal. We attend to events, to Congress, and I want to take the opportunity to invite you. This year, we're going to be with Innopsys in FENS Forum in July in Barcelona, great city to visit. And we are going to present InnoQuant, the instrument that we are talking today. We are going to be in the booth 38, and you can bring your slide and scan it and maybe play a little bit with the instrument. As I said, Paralab Bio is one division of the big company called Paralab, but we are focused in technology for bioscience, in research and industry. Our main portfolio is focused in bioimaging techniques, bioprintings, animal imaging techniques, and instruments like PET, SPECT, CT, MRI, bioluminescence, and fluorescence imaging. Also, we have super resolution microscopy, scanning electron microscopy, and intravital microscopy. We have some instruments for cell counters, cell processing, flow cytometry, and some biophysics and molecular interactions, and instrument for live cell imaging and single cell tracking. During this webinar, we will explore how InnoQuant helps researchers simplify whole slide quantitative immunofluorescence workflow. We will see how the system reduces reliance on confocal microscopy for large slide screening and enable faster time to results without post-processing step. We also discuss how high reproducibility and quantitative reliability can be achieved thanks to 16 bits dynamic range and uniform field illumination. In addition, we will review how simultaneous four-color images and AI-guided tissue detection contribute to automated and workflow efficiency. Finally, we will explore how stitching bias can be minimized thanks to highly uniform image acquisition. Also, we are going to listen to Erik that from the customer experience. With that, I am delighted to hand over to our first speaker, Charline. The floor is yours.
So now, as you perfectly introduced, I'm going to share my screen and start talking about the InnoQuant slide scanner. So, please tell me if you can see the screen well. I think you should be able to see the presentation. Okay, so as Amarlig said, I am going to present the InnoQuant slide scanner. This is what we call a quantitative slide scanner, and we are whole slide imaging device, and our strong asset is that we are scanning line by line without tiling and stitching, and so without post-processing. And I will describe to you just how in this short presentation. So first, we can ask ourselves, what do you have at the moment? So what matters when you have histological slides? So of course, at the beginning of your workflow, you have slides with tissue, and in the end, you want to obtain robust, reproducible, and quantitative data from these tissues to further be analyzed. So a lot of different parameters matters during image acquisition to obtain this robust and reproducible data. And our slide scanner InnoQuant can really answer all of these parameters. So first, I will give you a very short introduction about how the technology works. So we are a confocal light slide scanner, and something that is very interesting is that we have 4 excitation lasers, so 4 excitation wavelengths, UV, 488, 561, and 640 nanometers. And our strong asset is that we are scanning these 4 colors simultaneously, like a short square on the sample. And this allows to scan the 4 colors at the same time, because then we will collect the 4 emission beams simultaneously also. And then the 4 emission beams are going to be spatially separated and directed towards their corresponding detector using a small pyramidal mirror, as you can see on the right part of the screen. The detected wavelengths are going to be spatially separated and directed towards their corresponding PMTs, which are our detectors. So here you have a more detailed drawing of the inside of the instrument. So we are using laser for excitation. And then, as I was just saying, we are also using PMTs as detectors. Yes, so our slide scanner has confocal capabilities, which are also enhanced by a small pinhole that we do have in front of our PMT. Yes, so we are scanning with our 4 lasers simultaneously and then collecting the emission beam, and each emission is detected by its right PMT. So what makes it different rather than other type of instruments is that we are scanning line after line. And so in regular slide scanner, usually the detector is a camera and not a PMT. And so we can scan field of view after field of view, and this creates this type of mosaic effect that you can see on the screen. So then the image needs to have a post-processing steps with stitching and shading correction. And then the image is saved. With InnoQuant, we are scanning line after line. And so the image is constructed on the full width. So there is no field of view like you can see on the first image. And so this creates no need for a post-processing steps. So once you have scanned your image, you have your raw data, and you are good to go. And this is very important, especially when you want to do some image analysis and quantify or use a threshold to detect some positive objects, because this way you can be sure that the intensity of the pixel that you are analyzing is the raw value and there has not been any post-processing step, which is very important for use of threshold, because usually when we use a threshold, we decide on a positive value, and all pixels that are above this value are considered positive, but we need to make sure that these pixels are exactly comparable from one part of the image to another. So here I have a short example of a brain section, and as you can see, we can really not see this field of view effect and this mosaic effect. And this allows us also to have a very black background without any noise in the background. So we can see that we have the glioblastoma here on this mouse brain, and around it are located some astrocytes. And here the researcher wanted to check the distribution of astrocytes located around the tumor. So they really needed to be able to know the level of intensity of astrocyte cell on the full slide. So as I was saying, we are looking like a confocal microscope because we have lasers and PMTs, but what is very different is that we are a slide scanner. So this is an automated instrument, and InnoQuant really fits throughput and speed that you can expect from a slide scanner. So first we have a 24 slide autoloader capacity, and then so you can load up to 24 slides. You can detect automatically all the tissues that are on your slide, so brain section. In neuroscience, most of the time, the focus is also fully automated. And for also a very efficient workflow, you can pre-save some configuration and just apply the parameters to all your batch and all your experiments. And for each experiment that you will conduct, you can have a pre-saved configuration, which allows you to save a lot of time. So you can put all type of different sample at the same time, and then have different settings for each slide also, or have the same setting for all the slides. And this allows for very robust batch scanning and with automated tissue detection to really being able to scan in the most automated and efficient way as possible. Here on this example, you can see on the left of the screen that on the slide there were 12 brain sections. This is actually from Translucence Biosystems. And so you can see that we were able to scan all the brain sections one after the other in a fully automated way with the same parameters that are applied on each section and with an automated focus. So we also ensure very quantitative measurements because we have a quality control package in our system. So it comes with an Argolite slide, which is a lifetime warranty quality control slide. And we have built in what we call a check scanner procedure. This procedure is to be around like once a month or as often as you wish to check that your scanner is in the same state of parameters as when it left our manufacturing premises here in France. So you can check, for example, the intensity of the laser, linearity of the PMTs, the crosstalk between each channel. And this allows for you to make sure that each experiment can be comparable not only from one slide to another, but also from one day to another. And also if you have different laboratories, different locations from one day to another. Just checking that we can still see. Okay, sorry. So here I'm just going to give you a few examples to see how now this instrument can really support your research in neurosciences. So we have gathered opinions and samples from a lot of researchers in the field of neuroscience, but different applications. For example, Jean-Christophe Delpech in France is working on how nutrition and affects the brain, and he is also working on age-associated disorder. And for him, InnoQuant is really the perfect tool for accelerating the time to results for his central nervous system cell quantification, because he can scan in an automated manner all the data, and it takes approximately 10 minutes for a mouse brain to be scanned with the 4 colors at the same time. So for him, compared to using a white film microscope, it was really faster, and he really appreciates it. Dr. Oana Chauvert, she works on the glioblastoma, so she was the researcher who provided this nice brain slide that I just showed you. And for her, InnoQuant really has a bit the same image quality as other type of microscope or scanner that use an algorithm to enhance the quality of their image. So it's really interesting because without any post-processing, you can have an image as good as an image that has been modified. So it means that you can really trust the raw data to obtain really robust results in your analysis. Dr. Bruno Benitez in Boston is working on Alzheimer's disease, and what he really also loved about this scanner is that he can just launch all the batch, and then the batch will run itself, and then he can free up the operator. So here we have a short case study. So he really needed fast scanning and automation because he has a lot of slides with so many brain sections on each slide. And so he was also having trouble with the focus on his slide. And so he really needed to distribute each focus point on each brain slide, and it was taking forever. So he was also looking for a really reliable focus. So before implementation, he was spending approximately 5 minutes for scanning one brain section and 5 minutes for the stitching. So for 16 sections on one slide, it would take up to 8 hours. And because he needed to distribute all the focus point, etc., he had an operator full-time at the scanner. But with the InnoQuant, you can slide one brain section in 8 minutes and the full slide in approximately 1 hour 40 minutes, focus included. And this can be applied to 24 slides in the scanner. So the operator during all this time is free for other lab experiments. So this research lab was really able to accelerate also all the scanning for their brain sample and to have a robust analysis because of the quality of the intensity. And the last example, I will not discuss into too much detail, is the one that we have conducted with Translucence Biosystems. So Erik Castillo will probably discuss this a little bit more right after me. But we have conducted a short study with them on neuroinflammation and tried to check the influence of LPS on the inflammation of microglia. And the data from InnoQuant were really reproducible, and so it could accelerate the research and development progress. And then because also of this quality control that I described a little bit earlier, it can really be a robust and a very trustful data also for regulatory grade documentation. Okay, so thank you for listening to me, and I'm really sorry again about the small internet shutdown that I had. I am now done with the technical presentation of the InnoQuant, and I will give the floor to Erik Castillo from Translucence Biosystems for a presentation of his company and of data.
Okay, let me go. Let me go ahead and share my screen. Can everybody hear me?
Yes.
Can anybody see my presentation? Okay, so thanks everyone for perfect. Thanks everyone for joining today. My name is Erik Castillo, and I'm a senior project manager at Translucence Biosystems. I'm really excited to be presenting on whole slide quantification made simple. What I'm going to show you today is a complete automated workflow from slide scanning all the way to publication-ready quantitative data that we think can genuinely transform how your lab approaches immunofluorescence on brain sections. Let's get to it. Here's what we'll cover today. So we'll start with some background on what Translucence does. We'll walk through our 3D platforms briefly, then spend the bulk of our time on 2D quantification workflows and the InnoQuant collaboration we did. That's where the real exciting data lives. So Translucence Biosystem is built around one core idea, and that's making 3D histology of the brain accessible and quantifiable at scale. We combine tissue clearing, light sheet imaging, and computational reconstruction and machine learning-based imaging analysis into a single end-to-end platform. We were actually recognized at the 2020 NIH Brain Initiative Investigators Meeting and won first place in their video contest. And I just want to quickly play you a quick video of what these 3D histology can look like. In here, what we're showing is a cleared mouse brain stained with autofluorescence of GFP. And what you can see is that with 3D histology and light sheet microscopy, we can actually image the whole brain fully intact and span through it to see not only cell bodies, but as well as their projections, and also retain all of the anatomical positions of those cells. Now, to understand why this matters, you need to appreciate the fundamental problem. Tissue is opaque. Traditionally, to get any information out of a brain, you have to slice it hundreds of sections, each mounted, stained, imaged, and stitched back together. That's weeks of work, and you still get an incomplete picture. Tissue clearing changes that. Using iDisco or similar methods, we can turn an entire mouse brain transparent. What you're seeing here on the right is a real brain before and after clearing. And basically with the clearing protocol, very similar to what we do in immunohistochemistry, is you can go through a series of washes that removes the lipids and gets us to what we see on the left, on the right image, which is a transparent brain. And with the combination of antibodies and immunofluorescent dyes, we're able to then use a light sheet microscope to image the brain and get similar videos to what I just showed. But now let's talk about the quantification side of 3D histology. So this is Translucent Service Platform. We have 4 stages: tissue clearing, light sheet imaging, pre-processing, and quantification. The first 3 steps are moderate in complexity, although we've streamlined them quite significantly. But quantification, that historically has been the hard part. That's where project stalls, where variability creeps in, and where you need a specialized expertise. That's exactly where we focus our engineering efforts. And importantly, this isn't just a research tool. We offer this as a full pipeline service to CNS drug discovery companies, as well as a modular service if you just need help at any stage. Whether you're a pharma team that wants to handle samples and receive a data table, or an academic lab that just wants to jump in at certain steps, like do the imaging yourself and us do the quantification, we can support both services. And just quickly to go over our 3D voxel quantification pipeline, what ends up happening is that we can take raw TIFFs coming from a light sheet microscope. In parallel, we can run them through machine learning based cell classifications, and as well as do atlas registration to a reference atlas. And then the machine learning steps takes around 4.5 hours, and it's fully automated with no human in the loop, as well as atlas warping adds another 30 minutes. At the end, it comes together to give us a regionalized data. Every detected cell mapped to a specific brain region with intensity, density, and location metrics all ready for statistical analysis. This is the engine that powers everything we do, and as you'll see shortly, we've now extended the same engine to work on 2D slide scanner data. Here's a real example of what this pipeline delivers. We're looking at MPAS4 expression in immediate early gene across the entire mouse brain at 5 time points after light exposure: 0, 15, 30, 60, and 120 minutes. On the left, you can see the signal propagating through visual cortex and hippocampus over time. On the right, a volcano plot showing which brain regions are most significantly upregulated: primary visual areas, retrosplenial cortex, CA1 of the hippocampus, all biologically exactly what you expect in this study. The point here is the level of brain-wide region-specific time-resolved quantification used to require months of analysis. With voxels, it's automated, reproducible, and scalable. Now let's shift to the part that's most relevant to today's webinar, which is the 2D quantification and our collaboration with Innopsys. So this is a white paper we published together with Innopsys. The core question we sought to answer was, can a slide scanner-based workflow match the performance of a traditional wide-field microscope for quantitative IHC while being dramatically faster and more scalable? Spoiler, yes, and we're going to walk through the data that proves it. Here's a problem we're solving. 2D immunohistochemistry is standard in preclinical safety and efficacy studies. You want to ask, is there neuroinflammation? Is my drug affecting glial activation? These are all critical safety signals in CNS drug discovery. The images on the left show exactly what we generated for this study that we mentioned earlier. We stained for an LPS model and stained for DAPI, GFAP, IBO1, and CD68. On the top, we have wide view images as well as zoomed in and machine learned generated labels, which is all beautiful data, but the challenge is getting there with a traditional microscope. Traditional acquisition is done one slide at a time. You load the slide, you set up the runs, you acquire the image, you unload the slide, then repeat with the scientists involved at each step. In contrast with the slide scanner, like the InnoQuant, we can preload up to 24 slides and let the system run over time, freeing up scientists and enabling around-the-clock imaging. In addition, wide-field microscope imaging requires child acquisitions and downstream stitching. For a study with 20 or 40 animals, multiple brain levels, and 4 channels, that quickly becomes a major bottleneck. It simply does not scale, and that's a specific problem the InnoQuant integration has helped us solve. So very briefly, going going over the PMT slide scanner is Translucence partnered with Innopsys to integrate their InnoQuant PMT-based slide scanner directly into our voxel platform. What makes InnoQuant different is the acquisition principle. It uses a single focusing lens that line scans across the full width of the slide. No tiles, no stitching for lasers. Excitation lines are fired simultaneously, and 4 detected PMT detectors capture the emission signals through a pyramid beam splitter. You get 4 perfectly co-registered channels in a single pass. The impact has been significant. We went from averaging a project turnaround of 6 to 8 weeks down to 3 to 5 weeks, roughly a 40% reduction, while maintaining cellular level accuracy across all 4 channels, which is DAPI, GFAP, IVA1, and CD68 for this particular study. And the scanner can handle up to 24 slides per run. If you're running large cohorts, that changes everything. Now let's look at the actual benchmark data. We used an LPS-induced neuroinflammation model as well, a well-validated system where LPS treatment activates microglia, which you can detect with IVA1. On the left, the bar chart shows signal-to-background ratios for PBS control versus LPS-treated animals. The top row is InnoQuant. The bottom row is a traditional wide-field microscope. Both systems clearly detect the LPS effect. Both show elevated IVA1 signals in the LPS group. On the right, the raw images tell the same story visually. The LPS sections are noticeably brighter, more activated microglia throughout on both systems. The critical finding here, with the InnoQuant, we achieve comparable signal discrimination to the traditional wide field microscopes, but without the tiling overhead, without the stitching artifact, and at the fraction of the hands-on time. This is the validation that gives us the confidence to switch from using a traditional microscope to a slide scanner. Now that we can achieve high throughput imaging at scale, the next step is quantification, and that is where our voxel ecosystem comes in. Although voxels was originally developed for 3D light sheet datasets made up of terabytes of data, we have adapted the same computational infrastructure for our 2D histology projects by combining a GUI-based workflow builder, a run coordinator that queues jobs across Kubernetes computer clusters, a data browser, and integrated views such as ImageJ and Amarlig. We can quantify thousands of images with minimal user input. In the next slides, I'll highlight some of these key features of the voxel platform that enables these workflows. For 2D slice analysis, specifically, the pipeline is streamlined. What we can do is take a raw TIFF from the scanner, go through pre-processing, that then machine learning based segmentation can be performed on those images, and we can then fine-tune those outputs with deterministic filters. And what ends up returning from the pipeline is actionable data tables, no manual counting, no job queuing, unbiased, and most importantly, fully automated. This right here is our Maverick tool, which allows scientists who wants to have direct control over machine learning workflow developments. It's built on the Amarlig, it's user-friendly, multi-threaded, and designed for rapid iteration. Scientists can load up images, train models, and generate probability masks that can be visualized as heat maps. So here on the very far left, we have the raw image, and what the scientists can do is tell the machine learning models which signal is of interest, as well as identify what signal is not of interest. And then what gets outputted is a probability mask, which is on the next image. And what we can see is that we can actually visualize the probabilities by heat map, with the highest probability of it being the signal of interest being in red, and then the dark blues or the blacks being the lowest probability. We can then also go even further and add a label filter, which actually can go through all these probabilities and filter out signals that do not meet certain thresholds, and ultimately fine-tuning so that we get label masks. And here's just sort of a visual idea of what the different machine learning-based quantification can look like. A traditional workflow that can be done with a standard thresholding can do a pretty good job of picking up signal of interest that has a high discrimination between the background. But what it can do sometimes is notice the difference between artifacts. So on the left is a standard threshold model where you can see that there's folds that are still being picked up by these thresholding parameters. But when you use Voxel's machine learning quantification, on the right, you can see that the folds are no longer being picked up and completely avoided, while still retaining the signal that's underneath those folds on both the right of the tissue and the left of the tissue, which allows the data to be cleaner and also makes us more confident that when there are artifacts, they are being avoided by the machine learning algorithms. Now, to take that together, for teams that want to build their own analysis pipeline, we also have our workflow builders that are all GUI based and nothing requires coding. And what this allows us to do is that it's a drag and drop workflow builder that covers 2D slices, 3D atlas registration, and AI segmentation and statistical analysis. You can design the workflow once and run the coordinator and execute it across the entire data set. So what we have here, oops. So we have here is an example on the left, which shows that you can have images in a project folder, and on the middle is build out workflows that can take your entire data set and run it through the same quantification pipeline so that you know every single image and every single data point runs through the exact same parameters. And this is all done automatically through these workflow builders that allows scientists to be hands-free as well as reduce bias by making sure all of your data is having consistent standings across the board. And of course, with all this high throughput and automation, one key thing that we still want to focus on is QCing. So one thing that we've also added part of the pipeline is called Stitch View, which can take all of the images in a data project and turn them into bite-sized PDF files that can be downloaded into any computer, and then can be viewed and organized however you like. So for this particular case in this particular study, what we can actually do is align all our control animals in together and all of the LPS animals on together, and then you can see visually any qualitative differences between the treatment groups, and even within a certain treatment group, we can see whether a particular sample falls in line with the expected treatment groups, as well as just visually see any artifacts so that we can flag them downstream. And here's the bottom line. Here's the bottom line output: 4 markers, 4 bar graphs, real statistics. DAPI is stable across groups as expected, which serves as a good internal control. For the GFAP signal, it shows a trend, but IBA1 and CD60, which are all microglia activated markers, both reach statistical significance with p equals 0.05 and less than 0.001 respectively. LPS induced neuroinflammation, and this pipeline detects it cleanly, reproducibly, and automatically. This is the kind of data you can put in a regulatory package or a paper. And here's something that we're still working on and trying to finalize is a regional analysis tool, which we're now taking our raw images and aligning them to a reference atlas, very similar to what we did for the 3D platform. However, for 2D, it does get a little bit more complicated just because 2D slice histology a lot of times introduces a lot of variability within sections across animals because of just the method of cutting sections. And so then it still requires a bit of manual tuning. And so we're still trying to fully automate this as well. But so far, what we've been able to achieve is still regionalized data. And here is an example from this same study that showed a significant difference in certain regions like the brainstem, the midbrain, the isocortex, and the hippocampus formation. So to wrap it up, what we've shown you today is a validated end-to-end workflow with using the InnoQuant scanner for fast artifact-free host-like acquisition combined with voxels, machine learning-powered analysis for automated atlas-registered quantification. It cuts projects turnaround time in half, scales to large cohorts, and delivers statistically robust regional-specific data. If this is relevant for your work, for the work you're doing, whether that's neuroinflammation, safety histology, or any multiplex IHC studies, we'd love to talk. Thank you.
Thank you, Eric. Thank you, Charline. Now, Q&A question. So I have one from here. Uh, how easy is to integrate the system into your workflow, Eric?
Sorry, can you repeat that one more time?
How easy was it to integrate the system in your existing workflow?
Yeah, it was the InnoQuant system.
Yes, yes.
Yeah, so it was actually it's actually quite simple once it's done. So it does require at the front end, it does require our software developers to look at the data and see how to parse it into the system. Because for us, one of the key benefits of slide scanner data is that everything is contained within one data file. So it means as far as like logistically, our workflows are able to just take that one data file that contains all of the metadata, which is multiple channels, multiple scenes, and as well as multiple resolutions. And so then what that allows us to do is uh, have scripts behind the scenes that can take that one single image and just repeat that across all of them. And we get to choose what channels we're interested in for for a particular study, as well as choose the resolution we want, and then also split them up so that instead of having because before with a traditional microscope, it outputs every single section as one image, and then every single channel is one image. Now, 12 samples becomes it can become up to like 240 samples depending how many sections. Here, those 12 samples are still 12 individual files, and then once they go into our system, the system can split everything apart, keep everything organized. But for the scientists, you don't even actually have to take a look at that because everything is done streamlined. And at the end, we just get the data tables. But of course, you can always go back and individually check those data files.
Great,
Thank you.
great. Which another application can we can use the InnoQuant for Charlene?
Yes, so first, just before answering your question, I wanted to say something about what Eric said, that we have one metadata file with multiple channels and multiple resolution, because what we deliver is a TIFF file, so it's an open source file that you can use with any analysis, and it's pyramidal, which is why there are different levels of resolution, and you can in your only one image, one scan, you have the full view of, for example, here your brain, and then when you zoom into the image, you also have the cellular resolution. And to answer your question, you can, so here during this webinar, we wanted to focus on the neuroscience, but of course it's a fluorescence scanner. So we can have applications in all different types of field, immunofluorescence, and also cancer research. So if you are studying also neurosciences, it can be glioblastoma. You can also have application in plant science, gene therapy, and all this type of field that require some either autofluorescence or immunofluorescence assay. Yes. and
Great,
and
great.
I
Sylvia,
could,
to add something.
yes, I just wanted to ask, I could see on the different polls that were uploaded that a lot of you are using the confocal microscope. And so I hope you could see how this scanner is actually really a confocal-like slide scanner. It's the only slide scanner on the market with PMTs as detectors. So I hope I could really give you enough information to understand how it can be like confocal like slide scanner. Sorry.
Okay, we have a question from Esther. How many micrometers the tissue slide has?
I think you mean the thickness of the tissues, and so usually it depends on the tissues. We have scanned tissues like 10 micron thickness up to 40 micron thickness, and we also have scanned tissues with 200 micron thickness. But for this, the tissue needs to be cleared, and so translucence has actually a very good clearing tissue device. So you can also clear your tissue and so image a little thicker tissue. But I think in between 5 microns and 40 to 50 microns, the system is completely able to scan your sample, because we have a depth of field of around 10 microns.
Thank you. Here is a question regarding to the if easy to use the instrument. They need a lot of training. What do you give us that information
Yes, very
about?
good question. So the installation of the system is actually really fast. It's quite plug and play. And the system is quite compact, doesn't need a dark room, and doesn't need an anti-vibration table. So you can really install it in your lab. And for the training, it's very easy to use. We really want our workflow to be as user-friendly as possible. So usually 2 hours of training is already enough to be able to understand how to manage the software and the scanner. And yes, with this training, you're good to go. And of course, then we can have a little more in-depth training to really create some configuration that the user can reuse and have already everything pre-installed into the scanner.
Okay. There is a question from Esther. I don't know if you can read it. So,
You cannot do a Z-stack, but perform staining of sequential slides and then 3D them. So with the InnoQuant, we are very dedicated to 2D imaging. So we have a robust focus algorithm that will find the focus. Then we have the possibility with the hardware to do different Z scanning. But at the moment, we cannot reconstruct the 3D image. However, Erik presented a few 3D workflow because at Translucence, they are also specialized in 3D analysis. But for the InnoQuant itself, you can really focus on 2D. I hope that was a good answer to your question.
Esther, did we answer? Okay, perfect. Okay, to Erik, would you recommend this system to other labs and give us two whys you are recommending this system?
Yeah, definitely. I mean, this has really helped us keep up with the demand of our projects, and also what's really nice too. Like what I think one of the main benefits is the images straight from the slide scanner look very pretty. There's no stitching involved, and sometimes, especially when you're trying to show like, I think pictures are worth a million words, and so if you can show a beautiful picture and really shows the difference between difference between treatment groups because there's a high standard background ratios and so forth. And it being just straight from the scope makes my job a lot easier. And also just the amount of hands-free that it's able to give our scientists allows us to run multiple projects in parallel. So for sure, I would definitely recommend it.
Thank you. From Esther, also, is it also possible to perform colocalization analysis with 20x resolution?
Yes, yes. So actually, as I said, the system can go down to 0.5 microns per pixel. So this gives the equivalent of a 20x resolution. And you can definitely perform colocalization analysis. This is something that we have already done, either on brain sections, but also for other applications like cancer. And you can really be sure of the colocalization because we don't have any crosstalk because of our optical design and also our QC quality control that will check the absence of crosstalk. And so you can be sure that two signals that are colocalized are completely from different stainings and dyes. So to answer in a more short way, yes, it is possible.
And we've done it as well. And like Charlie mentioned, because of the unique system, there's actually some microscopes that between the different epitopes, there's a little drift between them. And so when you try to colocalize them, you have to mathematically adjust them. But with this PMT slide scanner, that isn't necessary. And so it's actually made our job easier as well for colocalization analysis.
Okay, thank you.
Thank you for your testimony.
Yes. So I don't see more question in the chat. Maybe we can wait like what cost does it have? Well, we need to do a quotation. Esther, I think you are from Spain, so we can contact you and do some quotation.
Yes, you can contact Paralab Spain or Portugal if you are located in Spain or Portugal to obtain this information. And also, I have seen in the poll that some of you are going to attend the FENS conference, so we can definitely meet. And if you'd like also to obtain personalized demo or test the system with your own sample, you can contact Paralab or ourselves after this webinar, and we can definitely plan to meet at FENS or to organize a demo or to
Yes.
scan your slide.
Yes, I will remember that we are going to be on fence in a booth 38. You can bring your slide and we can scan, right?
Yes,
We're going to have the InnoQuant there, so you can play with the instrument. So, well, I don't know if there is any questions.
I
Okay.
think we have answered all the questions.
Okay. Well, I want to thank you, Eric, because he wake up very early because he is located in the United States. And Charlene, also thank you for your help.
Of
Uh,
course, thank you, and thank you, Eric, and to you, Amarlig.
thank you. Thank you to everyone. Well, we are here to serve. So see you later. Bye.
Thank you for attending.
Bye.
Goodbye.
Bye.